Everyone, thank you for being here. So our first talk in this sections is by Maggie Jones and Gloria Huang. It's about biosystem balancer, ecosystem in interaction simulation. And so over to you, Maggie and Gloria. Thank you.
Okay, hi, can you see my screen?
Yes, yes.
Okay, cool. Well, good morning. My name is Gloria Huang, and today I'll be presenting with my partner, Maggie Jones. Our project is called Biosystem Balancer. Biosystem Balancer is an ecosystem simulation game and learning tool to model the predator-prey interactions within a forest species ecosystem. We created this Mathematica program as our final project of our computational problem-solving course last spring. Our project implements predator-prey interactions through a differential equation model consisting of 4 equations, one for each species in a food chain. Our program teaches the user about the predator-prey differential equations and how altering growth rates, decay rates, and interaction between species affects the health or population of other species in the ecosystem. The user's goal in our program is to find the correct starting populations for each of the 4 species to prevent overpopulation and or extinction in a species. Finding the correct starting populations would result in an ecosystem at equilibrium in which all 4 populations stay constant over time without fluctuations. Once equilibrium is reached, we would say our biosystem is balanced. In our presentation, we will first introduce the differential equations we use in our project. Then we will explain the details of the main components of our program. Next, we will explain the user's influence in the simulation and how the user would go through using our program. And lastly, we will explain how each of the main components of our project, integrating explanations of our physical code with how that code is implemented into running our program.
The differential equations we implemented are commonly referred to as predator-prey or Lotka-Volterra equations. These equations relate predator and prey populations over time as they interact with each other, meaning that the predator consumes the prey. This is a simple version of this model that relates 2 species. In these 2 equations, X represents the prey population, Y represents the predators, and T represents time. dx over dt represents the change in prey population, and dy over dt represents the change in predator population. In the prey equation, the first term represents the growth of the prey population, which is proportional to the current number of prey that exist in the ecosystem. Because if there's a larger population, there's a greater chance that the individual prey will interact and reproduce. The negative second term is the rate of predation in which the predator consumes the prey. This death rate is proportional to the interaction between the predator and the prey, which increases when either population increases. A larger number of individuals in general increases the chance that these 2 species will interact and prey will be eaten by the predator. In the predator equation, the negative second term represents the death rate of predators from a natural death. This rate is proportional to the current population of predators, meaning when there's a larger number of predators, the natural death number over a certain time period will be greater. The positive first term in this equation represents the growth of the predator population from consuming prey. When either population increases, there's a greater interaction between the 2 species, meaning the predator is allowed to eat more prey, allowing them to stay alive and reproduce. Here you can see an example ecosystem with coefficients plugged into the before-mentioned system of equations. The plots show the periodic behavior of the 2 populations over time, plotted with x being time and y being the species populations. The oscillations in this graph show the cyclic relationship between predator and prey populations.
The parametric plot represents the same ecosystem explained in the previous 2 slides. The plot shows the prey population as X and the predator population as Y, which can be used to better understand how their interaction causes a periodic population plot. First, the lack of predators causes the prey population to increase because they are reproducing at a rate higher than being eaten by the few remaining predators. The large prey population gives an ample food source for predators, which causes the predator population to rise. The high predator population then overfeeds on the prey, causing the prey population to decrease. But as the prey population declines, the predator population also starts to decline because of the decreased food supply, and this restarts the infinite cyclic pattern. Now we can apply that knowledge to this plot from before. The prey are plotted in red and the predators in blue, with their populations changing over time. First, the predator population is low, so the prey population can increase because they are being eaten less. Once the prey population is at a high level, the predator population has a large food source, so they can increase. This large predator population overfeeds on the prey, causing them to decrease. Then some predators starve due to the limited food source, and the cycle starts again.
Our differential equation model expands on the basic model just explained to relate the interactions of 4 species. We first added a carrying capacity term to the first equation to fix the unrealistic assumption that populations grow without bound and the ecosystem has unlimited resources. Adding this term resulted in increased stability to the solutions of our differential equation system. The first equation still includes a negative interaction term with the predator. The equation for the second and third species are similar in that they both include a natural death term, a positive interaction term from consuming prey, and a negative interaction term from being consumed by their predator. The fourth equation has a positive term from consuming prey and a negative term from a natural death. There is the absence of a negative interaction term because the fourth species is at the top of our food chain and thus has no predators. Now let's take a look at the way we transform this into mathematical code. Gloria, I think you have to maybe switch the screen share. So first we have the solve DEQs function that returns the solution plot for each species over time. We use the ND-solve function in Mathematica to solve the differential equation system we just discussed. We also added a condition to the differential equation system using the when event function. This built-in function acts to set the population of a species to 0 after the species dips below 2 individuals, because you need at least 2 animals for reproduction, so if there are less than that, the species will die out from the ecosystem. We did this because we were having a problem where the species population would go to a very small decimal number instead of reaching 0. The next function we'll show is set values. This function sets all of the global variables used in the differential equation to our desired values using one of the set list of variable values from the all variable sets list above. This allows us to handle the large number of variables that are within our differential equation system. The variables set in this function include birth rate, death rate, and interaction rates, along with time periods and species starting populations. The final important function dealing with the differential equations is the find equilibrium function. This function uses NSolve to find the specific population for each species where the change in population is 0, or the population stays constant over time. These specific populations result in an ecosystem in equilibrium, rather a balanced biosystem. The user's goal in this game is to try to find the equilibrium populations. This function returns the one solution where none of the species are extinct, giving us our equilibrium population used in the game. The plot of the ecosystem at the equilibrium looks like this plot below that shows each species as a straight horizontal line, meaning that the population will stay constant over time rather than fluctuate.
And as we explained earlier, our project has 2 main objectives: to function as a learning tool and as a simulation game. As a learning tool, we want to teach the user about the differential equation system implemented in our program. This is achieved through diagrams and explanations of those diagrams. We also want to teach the user about the relationships between species and how increasing or decreasing the population of one species will cause a fluctuation in the population of a different species. And as a simulation game, we want the user to implement what they learned on our learning interface in order to achieve success in the game. The objectives of the game portion of our project are to realistically model how predators and prey interact in an ecosystem, and to perform an analysis of the results from solving the differential equations. The analysis is shown to the user at the end of each round of gameplay, as well as at the end of the game.
Here are some example food chains that we used for the different ecosystems in our project. The first group in the food chain are the producers. These are the plants that get their energy from the sun and introduce it into the ecosystem. The second group in the food chain are the primary consumers. These are animals that eat plants like herbivores. For example, an insect as shown in our desert food chain. The third group in the food chain are the secondary consumers. These are animals that eat the primary consumers, like how a lizard eats insects. The fourth and final group in the food chain are the tertiary consumers, or the apex predators. These are animals that eat the secondary consumers, like a desert bird eating a lizard. They're the highest level in our ecosystem, meaning they have no predators. In our program, we have created 4 of these food chains in different biomes: temperate deciduous forest, desert, tropical rainforest, and a marine environment. The user is able to select which biome is used in their simulation.
And in our project, the user has many influences. The user will first be able to learn about the predator-prey differential equations implemented in our program. The user will learn how changing the coefficients in our model affect the species populations. These coefficients represent birth, death, and interaction rates between species. This is taught through allowing the user to manipulate a plot where they are changing the coefficients. We hope the user will walk away from the learning interface with a deeper understanding of how all 4 species interact to create the ecosystem simulated in our project and how the differential equations relate to the actual ecological processes in an ecosystem. Next, the user will be able to alter the starting populations of each species at the beginning of each round in order to try to balance the ecosystem. This is achieved on the Change Population screen. Altering these populations will change the differential equation model and thus the resulting populations from solving the equations. And after each round, the user will view a current state report showing statistics of each species population during that round. This includes the maximum and minimum populations and the average time length of one cycle. This report will inform users of how they can alter their starting populations during the next round to achieve a more balanced ecosystem. And lastly, the user will be able to make choices that affect the entire game, such as the number of rounds and the biome.
Now we will take you through the key components of our project. This includes the learning interface, change population screen, current state report, and endgame report. We will start by giving you an explanation of what role the component plays in our project, then show the code in action, and lastly, explain a bit of the code behind the component. So first, we're going to start with the learning interface. So here's the opening screen of our game. If we click Learn, the user will be shown a screen that teaches about the predator-prey differential equations implemented in our program. Different diagrams of the differential equations are shown, along with explanations similar to the ones we gave earlier when teaching you about our differential equation system. The user can then click All Species to view a manipulation plot containing all 4 species. The user is able to change the time period shown on the graph, informing the user of how the predator-prey relationships develop as time progresses. An explanation is also given on this screen to explain the predator-prey relationship in more general terms. Then, the user can view a manipulation plot for each of the individual 4 species by clicking the different buttons at the top of the screen. Here, the user can manipulate starting populations, interaction rates between predator and prey, birth rates, death rates, and/or carrying capacity, depending on the species selected. Changing these values changes the solutions to the differential equations and thus the population of the species selected. Lastly, on the learning interface, the user can view the interaction plots between 2 adjacent species in our ecosystem by clicking on the second row of buttons. These pages show parametric plots that plot 2 species populations against each other. Here, the plots show the plot between the producer and the primary consumer.
We will now explain a little bit of the code behind the learning interface. Here's an example of one of the manipulatable plots on the learning interface. This specific plot is for the second species in our ecosystem. the primary consumer, or the herbivore. We use the manipulate function to constantly resolve the differential equation system and show the updated plot as the user changes the values associated with the variables in the system. As an example, if you increase the interaction with a predator species, this species will be kept at a much lower population because they are being eaten at a higher rate. And now we are going to transition into the gameplay. So after you are done exploring this learning interface, you can click back to home, and then this takes you, let me reshare that. So after you go back to the home screen, you can click play game, and then this takes you to the options screen where you can choose the number of rounds you wish to play and the biome used for the game. For this simulation, we will choose to play 3 rounds and then choose the marine biome. Maggie, I think you're mute.
Sorry. This brings us to the change population screen. The change population screen will be shown to the user at the start of every round played. The intention of this screen is to allow the user to change the starting populations of each species. Changing populations will change the solutions to the differential equations and thus change the resulting populations. There is one correct starting population value for each species that will result in an ecosystem in equilibrium, or rather a balanced biosystem. This value is what the user is trying to find on this screen. After adjusting populations and you're satisfied, you can click Calculate Populations, which will take you to the current state report. After each round, the user can view the current state report. Sorry. Gloria.
Yeah, so this current state report appears after the user is done changing the populations and clicks the button Calculate Populations. This screen informs the user of how their selected starting population affected the ending populations of each of the 4 species during that round. And in our program, a round signifies a certain time period. Thinking in real life terms, you could consider a round to represent one generation or a season. And for each species, a plot is shown, which shows how the species population fluctuated during that round. Underneath the plot, we inform the user of the highest population and lowest population attained during the last round, along with the average time length of each fluctuation, which is the period of the plot. Then at the very bottom is a suggestion to the user on how they could change the species population during the next round to become closer to the starting population that results in a balanced ecosystem.
Now we will show you some of the code behind this current state report. So this function, get min-max period times, is what we use to analyze the differential equations at different starting populations. So it helps us find the maximum population, minimum population, and average period length for a round played by the user. First, the differential equations are solved using the new starting populations that the user selected. We use the when event function explained earlier to append the time point where there is a local minimum or maximum in the function, which corresponds to the first derivative equaling 0. This function returns 3 lists, one with the times at which there are local minimums, one with the local maximum, and the time length of each period. This analysis of the differential equations is displayed on the current state report screen.
Now we are going to complete 2 more rounds of our game to reach the end and view the end of game report. So as you can see on this graph, we have to decrease the primary consumer, increase the secondary consumer, and then decrease the tertiary consumer. But the producer has already reached equilibrium, so we don't need to alter that population. So I'm going to click Change Populations. Oh, I don't know. I'm just going to restart this. Okay, so we're gonna redo that first round and then view the current state report again. So this time we need to increase and then keep the second one the same and then decrease. So I will do that on the next slide. So I will increase the producer, keep the primary consumer the same, then decrease the secondary and tertiary consumer to try to reach equilibrium. And then as we go to the second round, we will get another instruction screen on how we can better manipulate these populations. So now the first two populations have reached equilibrium, and now we need to decrease the secondary and increase the tertiary consumer. So let's do that on the last round to try to reach equilibrium. So I will decrease this and then increase the tertiary consumer. And now we did not win this game, but we got two populations in equilibrium, and now we can click view the results of my game to get to the end of game report.
This is the end of game report. The plot shows the final plot of all 4 species from our game and those same 4 species at their equilibrium starting populations. As you can see in the equilibrium plot, each species stays at a constant population. This screen also gives the user the correct equilibrium starting populations and how far off they were from those values during the game. That concludes our code demonstration. We hope you enjoyed our project and learned a little bit about these ecological interactions. Does anyone have any questions?
I'll just comment that I think it's a really cool project. That's neat to see the different species and how they stack up and how they affect each other.
Thank you.
Thank you.
Hi, so I was wondering, would it be easy to like expand on your model and like add a new biome like the way you guys made your project? Or would like that require a lot of extra work?
Yeah, so it would be pretty simple to add a new biome. We would just have to choose the new species for the 4 levels in the ecosystem for that biome and add it into the interface. We also could add like a 5th species by adding another one of the middle equations, which are the same format. So the producer is different and the apex predator are different, but the species in the middle are similar, so we could add a 5th or 6th, but it would get to be a pretty complex differential equation system. But yeah, there is a way to implement a more complex way.
Thank you.
Okay. Well, it's a nice project. And how about, have you ever seen about if there are several species, their population are evolving during a specific time, right? A time period. And then the proportions of their different species will change over time, right? And you know, in ecology, the diversity is a very interesting topic. The diversity of this ecological system. It would be a very interesting project to see how the diversity evolves over time under your model. So basically, you construct a diversity measure for at a specific at a time t, right? And see how this diversity change over time, you know, based on the model. It would be an interesting project if, you know, yeah, that's something come to my mind. I think that's a very interesting talk.
Yeah, thank you. We will definitely take that into consideration. We thought about working with Dr. Larry Alice from the biology department to make this realistic. So we can definitely try to continue that and maybe work on it more this semester.
Yeah, very, very interesting. Thank you.
All right. Thank you for your nice talk. And all right, our next talk gonna be at 10:15. And so we have about 5 minutes for break.
So our next speakers are Gloria Wang and Diksha Satish, and they're going to talk about employing mixed effects modeling and parameter space geometry in a differential equation model to analyze the healing response of diabetic foot ulcers with individual patients. Go ahead, Gloria.
Okay, hi. Yeah, my name is Gloria Huang.
And my name is Disha Satish, and our research mentor was Dr. Richard Shugart. And as mentioned, our research is on employing mixed effects modeling and parameter space geometry in a differential equation model to analyze the healing response of chronic wounds.
So first, I'm going to go into the background information on wound healing. So the onset of a wound to the dermal layer of the skin requires a well-orchestrated interaction of biological and molecular events in order to effectively heal the damaged area. These wounds can be categorized as either acute or chronic with respect to healing. Typically, acute wounds progress through 4 systematic stages during the wound healing process: hemostasis, inflammation, proliferation, and remodeling. Chronic wounds, on the other hand, have an extended stay in the inflammatory stage. This causes the unpredictable nature in the healing of chronic wounds and consequently causes a problem in treatment to healthcare professionals. Because this wound healing process is very sensitive, healing must be precisely regulated for optimal healing. Any perturbations to this process can transform an acute wound into a chronic wound. One type of chronic wound is the diabetic foot ulcer, which has an enormous impact in the overall global disease burden because of the increasing prevalence of the disease. Our research is aiming to combat this unpredictable nature through analyzing a mathematical model that investigates how 4 vital state variables are interconnected at the wound site.
There are 4 state variables included in this mathematical model: matrix metalloproteinases, or MMP1, tissue inhibitors of metalloproteinases, or TIMP1, the extracellular matrix, or ECM, and fibroblasts. The ECM makes up the lower layer of the dermis. This is where collagen production occurs. ECM itself is a connective tissue made up of collagen. When this tissue is damaged, enzymes called matrix metalloproteinases break it down to allow for the production of new ECM. This allows fibroblasts to migrate to the damaged tissue. Fibroblasts are the primary mesenchymal cells present in the wound, and they produce collagen. TIMPs are responsible for regulating the activity of MMPs, which in turn regulates the degradation of ECM. It is crucial that MMPs are regulated well since they are integral in the synthesis of ECM. The interactions between MMPs, TIMPs, the ECM, and fibroblasts is crucial to the wound healing process.
The mathematical model for this research consists of 4 differential equations that represent each of these 4 state variables with respect to time. The importance of these 4 variables is rooted from the extended stay of chronic wounds in the inflammatory stage. This causes a very high level of MMPs and consequently causes the ECM to be degraded faster than fibroblasts are able to construct it. This imbalance requires that TIMPs should carefully control the level of MMPs to maintain balance in the ECM production.
So the MMP1 fibroblast production is modeled by a Hill function of order 3. MMPs are assumed to degrade linearly with K3 as the rate constant. In wound healing, TIMPs inhibit MMPs by binding to the enzyme in a 1-to-1 ratio. This regulatory effect is represented as an inhibition term by the rate constant K4, which is proportional to the MMP and TIMP concentrations. And that's our first equation here. The second equation is for TIMP1. It is also modeled by a Hill function of order 3. It is assumed the fibroblasts will produce TIMPs if MMPs are present. The same inhibition term used in the MMP equation is also used here, since the binding between the TIMPs and MMPs is generally irreversible. The rate constant K7 represents a linear degradation of the protein to yield the final TIMP equation. So the third equation is for the ECM. And so the fibroblast production of ECM is modeled as a constant rate with a carrying capacity. In addition, the ECM degradation from MMPs and the natural decay of ECM are also represented as constant rates. And so fibroblasts are included in this model as cells that produce MMP1, TIMP1, and ECM. They are represented using logistic growth with carrying capacity. The cell death rate is also modeled here as a constant rate. And so that's the last equation.
And in these four equations are 12 parameters that represent different entities in the healing process, such as cell and protein growth rates, decay rates, and interactions. These parameters are the main focus of this research. The data points used in the analysis of the model come from a study conducted to measure the levels of MMPs, TIMPs, and ECM in the wound fluid of 16 type 2 diabetic patients over a 12-week period. The goal of the study was to describe changes in MMP and TIMP levels during the healing of diabetic foot ulcers in order to find possible predictors of healing. The authors of this study took measurements of the percent wound closure, MMP1, MMP2, MMP8, and 9, and TIMP1 at the beginning of the study, week 1, 2, 4, 8, and 12. It was found that the MMP1 TIMP1 ratio is a predictor of wound healing in diabetic foot ulcers, which is why the mathematical model focuses on these two factors.
So mixed effects modeling was utilized to generate parameters for the individual patient data. Mixed effects combines the individual and population parameters for each patient. So the population parameter represents an averaged response derived from combining the data for all 16 patients. And the individual parameters represent the individual patient response based on each patient's separate data. Thus, it is unique for each patient. So mixed effects modeling was implemented in Monolix, a modeling software created specifically for that purpose. The stochastic approximation expectation maximization algorithm, or SAEM, is Monolix's implementation of the stochastic algorithm. SAEM is an iterative algorithm that uses Markov chains to estimate population parameters. So after the original SAEM estimation, Monolix combines the resulting population parameter estimates with the individual patient data to determine the empirical Bayes estimates, or EBEs. So EBEs are the mode of each individual's conditional parameter distribution. Essentially, they are the most likely values for the individual parameter. So the data collected from all 16 patients were inputted into Monolix and run through the SAEM algorithm. So here are the individual results for the parameters K1 through F5 for all 16 patients. Using Monolix's SAEM algorithm and EBEs, all patients were able to be curve fit. Patient 16's MMP1 data had 0 values for both week 0 and week 4. The original patient data was only calculated to 2 decimal points. Thus, it was assumed that the 0 values could be replaced with 0.001 in order to curve fit the data using the mathematical model. The week 4 value was not considered in the curve fit. Patients 10 and 13 was unsuccessfully curve fit in the past due to a lack of data recorded for them. Both patients only had 2 data points for MMP1, TIMP1, and ECM. Mixed effects modeling using Monolix was able to bypass this issue since population parameters are calculated using data across all patients, from which the individual parameters are then derived using the EBEs. The minimal data was able to be compensated for. The results in Table 2 are population parameters generated from the SAEM algorithm from which the individual parameters were derived. The results from Table 3 indicate how crucial a parameter is to the overall healing process. Parameters with low standard deviations are more likely to be important to wound healing since there is a minimal oscillation between individual patients. For example, the parameters K1, K8, and K11
have lower standard deviations, so we can predict that these parameters are more critical during the healing process. So through Monolix, all 16 patients were successfully curve fit, and mixed effects modeling was able to circumvent the prior issues we had with modeling patients 10 and 13.
My research is further analyzing the effects of each of the parameters in the model to find which is most sensitive or influential. However, my research is not using the parameters generated from Monolix, but rather the original curve-fitted values. Finding these parameters will increase the predictive power of the model and therefore convey to healthcare professionals what should be a focus during wound healing treatment. So first, that brings into question parameter identifiability. We would know a parameter is sensitive if a small change in the parameter value causes a large change in the model output. A sensitive parameter in the model would indicate that the parameter has a strong impact on the actual biological wound healing system. This ties into parameter identifiability, which asks if a parameter can be uniquely determined. The lack of identifiability in a parameter would signal the insignificance of the parameter in both the model and the healing response. Thus, we are trying to see which parameters are identifiable and most sensitive. To determine the identifiability of the 12 parameters in the mathematical model, I have constructed three-dimensional graphs for each combination of two parameters in each of the 13 patients able to be curve fitted. This allowed the first parameter to be on the x-axis, the second on the y, and the sum of squared errors on the z-axis. The sum of squared errors allows us to see how the results from our differential equations compare to the actual patient data values, therefore telling us if the parameter tested is identifiable. The data points on the three-dimensional figure are the solutions to the differential equations. And we can conclude that the parameter tested is identifiable if there is not a significant difference between the minimum value on the graph and the actual parameter value. So now we talk about parameter space. For each graph construction, each parameter was assigned a parameter space or range in which it was allowed to vary. Growth parameters were originally allowed to vary from 0 to 200, while loss parameters were able to vary from 0 to 1. This ensured the range did not affect the solutions to the differential equations. And after each construction, the parameter was refined in predetermined increments of about 10 for growth parameters and 0.05 for loss parameters to better capture the unique geometry of each combination and patient. And because the graphs show parameter identifiability, a bowl-shaped graph with a clear minimum, as in figure 1, would allow us to conclude that both parameters tested are able to be identified. The minimum value on figure 1 closely matches the actual patient data value, signifying an insignificant difference and thus revealing identifiability. Conversely, a trough-shaped graph parallel to one axis, as in figure 2, would tell us that only one parameter is able to be identified. And lastly, a flat two-dimensional graph, as in figure 3, would tell us that neither parameter value can be identified. After constructing these three-dimensional graphs for all combinations in all patients, it was concluded that K4, 5, 6, and 7 were able to be identified most often and are therefore the most influential parameters in the model. These represent the interactions between MMPs and TIMPs, which was K4 and 5, the production of TIMPs, K6, and the natural decay of TIMPs, which was K7. All four of these parameters play a vital role in the maintenance of TIMPs, which is represented in the TIMP differential equation in the model. So all the results are displayed in this chart where you can see the four TIMP variables were able to be identified the most. And in the wound, TIMPs play the extremely important role of regulating the production of MMPs. MMP levels are crucial to ensuring the ECM isn't degraded too much or too little. Any change to the level of TIMPs will have a direct impact on the level of MMPs, which will then further impact the rate of ECM production. This is shown by the sensitivity of the TIMP variables. So this tells us that the TIMP variables are very important in the mathematical model. And to validate the geometry results, a parameter subset selection method was implemented to generate associated eigenvalues for each of the parameters in the model. So to illustrate the manner in which local sensitivity information can be employed for identifiability analysis, a Fisher information matrix was generated to find the associated eigenvalues for each of the parameters in the model. This subset selection method was implemented for various combinations of 2 parameters for each of the 13 patients. This allowed us to compare eigenvalue magnitudes and ratios of the 2 parameters tested to the results of the 3-dimensional graph construction, which also compared 2 parameters against each other. The eigenvalues generated from the parameter subset selection correspond to changes in the parameter values from the solving of the differential equation system, which allows the parameter value matrix to transform parameter space into its corresponding eigenvalues. Large eigenvalues correspond to larger changes in parameter values during solving, telling us the parameter is sensitive and thus identifiable. Therefore, this work aims to find which parameters have larger associated eigenvalues to conclude identifiability. So after running the subset selection for various combinations, I noted that each patient had varying eigenvalue magnitudes for each parameter. The parameters with the highest magnitudes were documented, and these results show that K3, 4, 5, 6, and 7 had eigenvalues with the largest magnitudes and are therefore the most identifiable. These results match the results of the 3-dimensional graph construction, except that K3 was also able to be identified. And moreover, a ratio of the eigenvalues from each combination was created and analyzed to predict the geometry of the resulting combination. A small ratio, which was about less than 20, is a likely indicator of a bowl-shaped geometry in which both parameters are able to be identified. Higher magnitude ratios indicated only one or neither parameter was able to be identified. And most eigenvalue ratios follow this pattern except for the combinations that formed an identifiable combination, meaning that the identifiability of one parameter is dependent on the other parameter. So together, the geometry and subset selection methods show that the maintenance of TIMPs are crucial in wound healing.
So in the future, we plan on using the parameters generated in Monolix in a local sensitivity analysis, which is similar to what Gloria is doing right now. And so this is in order to identify which parameters are most affect the individual patient's healing response. We also want to implement the Monolix parameters in practical identifiability.
Do we have any questions for our speakers?
I have a question. You might have already mentioned it, but I'm in a coffee shop and I might have missed it. But do the parameters also include like medications that the patients are on, or is it just the healing process?
It's just the healing process. They just represent different factors involved in the wound site.
Okay, that's really cool. I think that's like fascinating research because I think that could potentially help them learn, like if they could incorporate medications, which ones help at what times. So that's really fascinating.
I think the data itself, the data itself is of the healing response of wounds, and I think that the patients were being treated, were being treated as you were going along. From the data itself, you do have, at least indirectly, kind of some of the effects of treating the wounds there. But there isn't a direct correlation otherwise with the parameters. But that's something that could easily be incorporated in this going forward.
Okay. Okay. Very good project. Again, how about? Well, I'm very curious about the problem of overfitting in your project. You have like you have like many parameters, like 14 or 15 parameters, right? And the data you give here is about 14 patients over a period of several weeks. And I would recommend to conduct probably a cross validation about how your model fit can be generalized to other to others, right? You can do maybe a leave one out cross validation and see how the model can generalize to other patients. Because the data size is relatively small, so you want your model, the results you get is very impressive. Okay, these the magnitude of these different factors can affect the wound healing process. That's very impressive. What I would recommend is probably consider cross-validation step so that your model is evaluated and that shows how your model can be generalized to other patients who has not been used in this training process, in your model, right? So you want to build a model which can be evaluated and also generalized to others. I would say that's going to be something you may consider to improve this project, probably. Yeah.
Yeah, I think it's a great idea, Lucan. One of the things that we did to try to combat that, at least, basically you've got it's an over parameterized system with sparse data. And it's one of the reasons that we moved to actually trying to do the curve fitting with mixed, using mixed effects modeling. And so, it's been recommended by several people, and so, the work that's done there, is at least I think it seems to be at least an appropriate step, in that direction. And I think your idea of cross validating this, I think could be a good next step with some of this as well.
Yeah, I really like this work, done by them, and they are really very impressive and very good project. Yes.
The students are very talented. Yeah.
Yes, they are. They are. I can't believe that they can do such a great job. Of course, Richard, you have done a lot of work with them, so that's great.
Do we have any other questions? Okay, let's thank our speakers. I guess I didn't say thank you for the first time, but let's thank them. And then our next talk will be at 10:45.